So, dear colleagues, today is the day of our Kharkiv chemical seminar, and it's my pleasure to introduce Professor Jeremiah Gassensmith, a leading scientist in supramolecular chemistry, nanomedicine, and biomaterials. He's a professor in the Department of Chemistry and Biochemistry at the University of Texas at Dallas. His academic journey began with a bachelor's degree in chemistry with honors from Indiana University in 2003, followed by the PhD in chemistry from the University of Notre Dame in 2009, where he studied squarine dyes and anthracene microcycles. He then completed a postdoctoral fellowship at Northwestern University with Nobel laureate Sir Fraser Stoddart before joining the University of Texas at Dallas in 2013. His research group explores the intersection of chemistry, biology, and material science with three major topics: nanomedicine and vaccine delivery. His lab pioneered in the use of metal-organic frameworks to encapsulate proteins and vaccines. Still responsible for biomaterials, his team develops smart materials that respond to light or heat, enabling precise drug release and cellular targeting. And finally, molecular imaging. His contributions have been recognized with numerous honors. He is a Fellow of Royal Society of Chemistry, Alexander von Humboldt Scholar. He won the UT Dallas President's Teaching Excellence Award and other honors. In short, Professor Jeremiah Gassensmith is a scientist, educator, and innovator whose work bridges supramolecular chemistry, biomaterials, and nanotechnology. Please join me in welcoming him to share his insights today. And dear Professor Gassensmith, floor is yours.
Thank you very much for the introduction. I'm very happy to be able to talk and very happy to be able to support this seminar. What I'm going to talk to you today about is the title of my slide here is on bacterial infections, where to find them, how to fight them, and how to keep them from coming back. Um, and so, as some background, one of the more complicated bacterial infections, the type of complicated bacterial infections that we have to face, are intracellular bacterial infections. And so these sort of hide inside of cells and they evade immune recognition and antibiotics. And some common infections include salmonella, listeria, tuberculosis, and even E. coli. Pardon me. I'm going to actually switch this to, okay. Um, as I said, they evade diagnostics and antibiotics and immune responses, and this ultimately leads to chronic infections and poor outcomes. So tuberculosis being, this is ridiculous, being an example of one. And so essentially, because they're able to exploit host cells to evade detection, and they are very difficult to treat, and they can contribute to the recurring diseases that are associated with a lot of these intracellular infections, and they can put more than 1 billion people at risk worldwide. One of the most common intracellular infections that are not really discussed extensively are urinary tract infections. And in fact, about 40% of UTIs are recurrent, especially in postmenopausal women, where this can become life-threatening or even difficult to treat or impossible to treat. Um, and so approximately 11% of the population in total ends up with these recurrent infections, and about 50 to 60% of women in their lifetime will have at least one UTI, and at least in the United States, the medical expenditures are pretty enormous for these, about $1.6 billion a year. And sepsis, which results from your current urinary tract infection, is a significant cause of mortality. And the UTIs themselves account for a quarter of all the sepsis cases. And so this is obviously one of the most severe diseases you can get from a bacterial infection. It's very life-threatening. Essentially, how UTIs become recurrent and invade the bladder is an ongoing area of research. But one area that we do know is that they are able to evade uh, detection and elimination by entering into the cells of the bladder. And you can see from the scheme here that essentially E. coli becomes bound to the surface of the bladder and will invade the umbrella cells that line the surface of the bladder. Well, they'll then proliferate and form intracellular bacterial communities, and then they burst out once again to spread. And this keeps the infection going. Once it becomes completely intractable and antibiotics no longer work, and this happens more often than we'd like to admit, a procedure that's been developed to remove the bacteria is electrofulguration, and you can see this here. What you're seeing is essentially the electrocution of the bladder cell wall, followed by removal of the debris that is caused. And so essentially this it's a fairly medieval feeling method of treating urinary tract infection, and it is because it's one of the areas in which bacterial antibiotic resistance is particularly rampant, and once it becomes bad enough, then it becomes life-threatening. So the problem with that method of electrofulguration is, as you saw, is entirely guided by eye, essentially looking for inflamed tissue in the bladder and trying to essentially destroy the tissue to remove those infected cells and then vacuum them out. The urologist essentially has no idea actually where the infection is. And so what my group has done is we have developed a new probe, or at least we've taken an old probe and modified it to specifically target the exterior negatively charged cell membrane of a bacteria that invades a host cell. Now, in this particular case, the idea behind a targeted molecular probe is essentially you have some sort of targeting element that recognizes some element on the surface of the bacteria that's unique to bacteria, as well as a reporting component. And in our case, we're going to use a fluorescent dye. And you can see here that essentially two modalities of imaging that we're going to be looking at are fluorescence and MRI. I'm not going to talk too much about the MRI stuff, but fluorescence imaging of intracellular bacteria is an area we're interested in because it provides an opportunity for, in this case, urologists to more target and eliminate the bacteria that is growing inside the cells in the bladder that antibiotics and the immune system are not able to get to. Now, our approach is to use this very unique lipid on the surface of bacteria, at least gram-negative bacteria, called lipopolysaccharides. And in order to do that, what we want is a high affinity association with something unique, as well as complementary shape of the molecule, so they sort of fit together like a puzzle piece. And they should work within what I say is a specific pH window, but we need to keep in mind that urine pH can vary from acidic to alkaline. Disease states can make those pHs fairly extreme, and so the window of binding is actually pretty high. Although intracellularly, the pH is a bit more regulated than it is extracellularly, so that's an added complication. In the past, people have shown that the use of the bis-zinc dipicoylamine, the molecule you see here, is an NX5 mimic. And so this binds to phosphates fairly strongly. And as you can see from the lipopolysaccharide, that colorful little molecule to the right is anionic and covered in phosphates. And so if we were to design a warhead or a targeting element based on that, what we're interested in is a high I'm sorry, if we were to design something that were to be able to go into a cell, pass through the cellular membrane, the zinc DPA molecule you see that I've illustrated probably won't work. The zincs are fully exposed, they're very cationic, they're Lewis acids. And so, in order to get into a cell membrane, generally you want to follow these Lipinski rules of five, which is to say that the system or the full molecule should have a partition coefficient of log P of less than or equal to 5, which is to say it should partition pretty well into both an aqueous as well as a non-aqueous phases. So essentially move from the outside of the cell through the greasy cell membrane and into the interior. And in order to do that, you don't want a significant amount of hydrogen bond donors or acceptors, and the molecular weight tends to need to be fairly small, although that's not. These all have room for wiggle, right? You can adjust these. So what we did in order to improve the hydrophobicity of the targeting element is we added this oxyanion onto the central targeting core. And what that does is it brings the zinc closer together and sterically shields them so that they're more greasy now. So they're more hydrophobic, but they're still able to interact strongly with oxyanions. And so we created this system here, which you will see is almost charge neutral with the zinc and the oxygens, as well as some sulfate groups and extended peg chains. One thing you'll note here is the dye structure that's down on the lower part, the black structure here, is that it has a couple of pendant arms that are pegylated to sort of protect this interior's core. And so this is a size 7 analog, but the methylene bridge is actually sterically blocked. That makes it more difficult for the dye to aggregate and clump up and cause problems, this particular aspect. And so essentially what we have now is a dye molecule with a targeting moiety that is soluble in both water and dichloromethane, which is going to be important to partition into cells going from the extracellular to the intracellular environment. Most of this work was done by my graduate student, Shailendra here, who did a great job. And essentially what this slide shows is a fluorescent image of binding to bacteria. This bacteria is called E. coli CFT703. It is a septicemic strain of UTI, so it's pathogenic bacteria. And what Shailendra is showing here is that essentially you can co-stain the bacteria with host, and stain the cellular or nuclear material, and overlay that with the binding of the size 7, and essentially show that the dye stains the surface of the bacteria. And this is more of a close-up here of this to show really good spectral overlap between the host and the size 7 stain. Um, and we've also done this on E. faecalis and a couple other bacteria. So the probe is actually pretty broad. It can stain gram-negative as well as gram-positive. So obviously, uh, uh, gram-positive stains, they don't have LPS, but they do have other anionic phosphates that are surface exposed. And so this dye binds to those as well. And here is M. bovis. So this is BCG. So this is actually a mycobacteria. Mycobacteria also have surface exposed phosphates, and this binds to it as well. So BCG is a close relative of tuberculosis. It's actually the live vaccine version used to inoculate against tuberculosis. So it looks like we are able to stain mycobacteria as well. Now, to do a quick study to see if we're able to penetrate into cells, we constructed giant unilamellar vesicles. And so you can see them forming in the little movie on the upper left. And we made these because it's very easy to visualize. And so if we were to take these giant GUVs, and add our dye, allow it to incubate for a little while, and then wash them and spin them down and recollect them, we can very clearly see that the dye actually does cross through the membrane and enter into the vesicles. So this is again good evidence that we're able to penetrate through a bilayer pretty effectively, and it goes into the inside of the cells. Um, One thing that this slide doesn't show, though, is if you just allow these red dots on the far right, so you see that the dye is inside, eventually that dye will diffuse back out. So the dye isn't necessarily trapped inside the vesicle. Once it gets in there, it's free to diffuse in and out of the cell, at least in these particular cases. And so when we move to an intracellular infection model, using our dye. Essentially, what we do is you can see the timeline of infection up here is you have a infection at time 0 followed by a gentamicin treatment. And so what this gentamicin does is it kills all the extracellular bacteria. So anything that didn't get inside the cells is now killed. So now we're dealing with purely intracellular stains. And then we add what we call our backed view. And our backed view is that dye that I've been showing you. We allow that to incubate for 2 hours, and then we do some membrane staining and nuclear staining. And so what we can show here is that in intracellular infection, so these are bladder carcinoma cell lines 5637. It's an epithelial cell line that basically UTI 89 invades. And you can see back view is staining these little red dots. Those are the bacteria inside the cell. Um, and then here in the green, what we actually did was we transfected the UTI-89 with a GFP reporter. So we can see where the bacteria is because it's also fluorescent green, and we can co-localize it with the red. And of course, you have this DAPI stain that is, I'm sorry, Hoch stain that is a nuclear stain for the cell, as well as WGA, which is a membrane stain that just stains the outside of the bladder carcinoma. And you can see that in these cases, we have good spectral overlap of the intracellular bacteria. If you add no back view, of course, you don't have any red fluorescence. You just see the green of the bacteria. Some of them are extravasing or extracellular vesicles. And if you add back view but have no UTI, what you note is no staining at all. And so what we think is going on here is essentially the back view is allowed to enter into the cell. It finds nothing to bind to, nothing to stain, and diffuses back out. So this basically gives us a very selective probe for infected cells. And it also shows us where in the cell the bacterial infection is. And to further drive this point home to show that the infection is inside of the cell, here are some Z-stack videos that are animated, and we do this rotational thing to basically show on the left that we do have co-localized staining with the pink showing overlap between the GFP and the back view, and then of course some nuclear stains. And on the right here, we have uninfected cells where we did add back view, but there's no red fluorescence because nothing got stained. In this particular case, the green is a membrane stain and not the GFP reporter E. coli. Now, this is all very good in a dish. Well, I wanted to see how it would look in vivo. And in order to do that, we used an ex vivo imaging model in which we infected a mouse bladder with CFT073. So this is a live animal infection, and then did an intravesical injection. So we injected the dye directly into the bladder. I note that you can also inject it into the vein, and it does the same thing. And in group A, the mice, which were infected with UTI-89 and then injected with Backview, um, you can see the very bright fluorescence associated with the infected bladder. In group B, these were not infected and injected with Blackview, and you see some fluorescence. And it's important to point out in this particular case that urine is not sterile. The bladder does have a microbiome, and those bacteria can get stained, but obviously the amount of staining is significantly less, and those bacteria are not intracellular. So what we're essentially seeing is probably some reminiscent fluorescence from essentially the mice not having urinated out all the bacteria or the bacteria that are naturally associated with the bladder. You can see the dye injection and the sacrifice time are actually relatively close together, and we probably should have given them a bit more time to urinate everything out, but that's okay. Group C was not injected at all. It was infected, and you can see no fluorescence here because there's no bioluminescence in the spectral channel that this dye was used. Now, to make it very clear that we're not just generally staining the bladder, what we did then is did the same experiment, except we removed the bladder and stretched it out and basically splayed it so that we can actually see. Now, infections in the bladder, much like infections in other tissues, are not diffuse. They're localized into little pockets or spots in the bladder. And so what you see here, down here at the very bottom of the no infection is the full bladder, stained with DAPI. And then if we look at the infected component, we see a red infection component in the center panels. That correlates with a small component of the bladder tissue. And if we magnify in on that, we can see the bacteria interspersed through the cells. And we can actually look at the whole tissue. See if I can play this video. Yep. And if we look at the whole tissue by sectioning, we can see that the fluorescence is located within the tissue. And so essentially the bacteria or the dye is able to infiltrate into the tissue and stain the bacteria that's associated with the bladder. Now, what we're going to do next is, of course, we're interested in destroying as well as detecting, and we can obviously detect, but one thing that we're interested in is in photothermal and photodynamic therapy to essentially then eliminate this bacteria, keeping in mind that this bacteria, when a patient is treated with this, would presumably be completely antibiotic resistant. And so we're basically at a point where we want to spare the patient as much discomfort as possible. And so we've actually designed a little system that we're implementing now in the lab that has a small catheter that is a fiber optic cable that we'd insert into the bladder and then irradiate and increase the temperature of the infected areas exclusively to essentially kill the cells that are infected. And then they release the bacteria once they're dead, and then the patient essentially urinates them out, or we can use a non-selective, more toxic antibiotic that is otherwise unable to penetrate into the cells to get at these reservoirs. Now, that was detecting them. Now, how about preventing them? So, an issue in preventing them, of course, is we're talking about vaccinations and prophylactics. And in a lot of cases, bacterial infections have historically been very difficult to inoculate against, the infection specifically. We do have vaccines that inoculate against toxins that they produce, but actually stopping the infection itself is fairly complicated. And then there are some other logistical issues that are associated with vaccines, in particular, the time-consuming and costly cold chain, because vaccines, of course, have to be refrigerated, and and a lot of vaccines, about 25%, are actually destroyed accidentally, and failures of these cold chains and the losses are fairly staggering, about $34 billion just in the lost costs. By some estimates, about 80% of the cost of an average vaccine that WHO distributes is about, I say 80%, is associated with just keeping it cold from the place it was manufactured to the place it's distributed. So this is a fairly significant problem. Um, but more about the vaccines themselves is that typically the efficacy of live attenuated vaccines tend to be pretty good, although it's not true for tuberculosis. CBG is not a particularly effective vaccine against tuberculosis. But as you may know, the difficulty in vaccinating a person that has HIV, which is often where you'll find people who need to be vaccinated against tuberculosis, is difficult because you cannot give them a live attenuated bacteria because it will hurt them. So there are some safety concerns. Subunit whole cell vaccines or whole cell vaccines in which the bacteria is dead. These are obviously safer, but they tend to be weaker immune response to. They have shorter in vivo half-lives, and they're cleared out pretty quickly. And then finally, about vaccines, one thing that we're pretty cognizant of is that delivery of vaccines usually involves a needle. And this is very disconcerting for a lot of patients um, that require follow-up. And so if you have to have booster shots, you have to go back to a clinic, you have to get injected, and nobody likes to be injected. And I think a lot of resistance to vaccines and patient compliance is associated with the fact that they are injected and those injections are invasive and patients simply do not like them. And so we are interested in addressing all three of these issues, and we are interested in addressing it using a type of material called metal organic frameworks or covalent organic frameworks. And essentially, what metal organic frameworks are these interconnected systems that use metals and ligands to assemble into porous materials. And you may have heard about them. They won a Nobel Prize this year. They're actually in Stockholm collecting their little award for them right now. And covalent organic frameworks, which weren't mentioned, but they were also invented out of the same lab as Omar Yaghi, who initially described the MOFs. He also described these COF structures, which are large porous systems. Um, and the porosity is interesting, and we certainly use porosity, but you go about this in a slightly different way than trying to use the pores. Now, before I get into it, I do want to review a few things about some current vaccine technologies. And this little slide here shows a few things. in which you have various different kinds of vaccine types, the subunit and synthetic peptides. These tend to be weakly immunogenic and they require adjuvants in order to work. You also have recombinant vectors type systems. So these go in and infect a cell and produce DNA, and then that makes the epitope. And similar to how mRNA vaccines work, essentially the cells make the antigen and then the immune system responds. And then there are a couple other things. And I draw your attention to, I already discussed live attenuated and whole cell inactivated. And these two are areas that I'm particularly interested in. Um, about 8 different diseases still use live attenuated vaccines, including MMR, viscerella, and tuberculosis. Subunit vaccines, because they're small proteins, they are not easily recognized by the immune system. And in order to get the immune system to start working, they are usually combined with something that's very immunostimulatory, and very popular one is alum. So essentially aluminum oxide. It's a metal oxide, and metal oxides and metals in general tend to be fairly immunostimulatory. And so you can see where we're going with this, using metals as an approach to stimulate the immune system, combining them with metal organic frameworks, and introducing a concept called biomimetic mineralization using what's a metal organic framework called zeolitic imidazolate framework. Now, how this works is essentially we're going to grow a crystal onto the surface of the antigen, onto the surface of the vaccine. And to do that, we combine zinc metal ions, the scaffolds, or whatever it is that we are interested in coding. So plasmids, proteins, liposomes, bacteria, whatever you want to your vaccine construct. And then we add the 2-methylimidazole. So this is the ligand, and the ligand and the metal and the antigen are all going to combine together. And essentially, the antigens, a crystal is going to grow over the surface of the protein or liposome or DNA or plasmid or whatever, and encase them completely. And what you ultimately end up with is something that's actually thermodynamically stable. So if you imagine protein denaturation, that occurs through large global motion, makes you a third or a tertiary structure that expands, moves. But if they're wrapped in a crystal or really tightly confined, that motion becomes very difficult. They can't actually begin to sample free space. They can get hot, they can wiggle, but they can't denature. But one thing that's interesting about moths that is unique to other mineralizing approaches is that they are what's called kinetically labile. And so while metal organic frameworks are very thermodynamically stable, if you add a competitive ligand like EDTA or even low pH, a proton, the ligand becomes protonated in low pH and it dissociates, or the metal itself is grabbed by a chelator like EDTA. And when the metal is taken out, that's essentially removing one component of a polymer and the whole thing falls apart. It just essentially dissolves. And we call that process exfoliation. And so we can encapsulate, create crystalline captured materials, and then we can remove that thing and get exactly the material back out. And it's called biomimetic mineralization because it's drawn from a natural process. And so you can see nature has these very beautiful mineralized little um, uh, I'm having a moment. So you find these little guys in sand. The name of them, for some reason, isn't coming to me. But they're very beautiful, and they're silicates, and they grow and they protect the organism. Biomimetic mineralization is essentially a co-opting of that process by man. And so what this process that you see here is actually this is from our group. The inside of here, this little light line that you see down the TEMs of all of these things. These are tobacco mosaic virus. And so tobacco mosaic virus is a rod-shaped virus, and it's lighter in contrast. And on the outside is the moth, the metal organic framework. And you can see that is darker in contrast because it contains zinc. It's a larger metal atom. And so what we're doing is essentially growing a protective shell onto the bits of the sensitive parts of biology. Um, And we have shown that essentially we can store things and transport things at room temperature that would otherwise require refrigeration because of this confinement. But what we actually found was that originally we had thought that this would be a great cold chain remediation technique. You'd ship a vaccine, the end user would remove the shell, and then you'd essentially inject it. We wondered what would happen actually if you didn't remove them off. If you injected everything all together in a patient, or if it would actually improve the efficacy of the bacteria, right? Or the efficacy of the vaccine. And we decided to look at E. coli. So we looked at urinary tract infections for which there really is no effective vaccine. There's no effective vaccine for any E. coli infection, actually. Um, and so wouldn't it be great if you could have a vaccine that essentially prevented you from getting urinary tract infections completely? This way, you don't have to worry about antibiotic resistance, and you can um, continue and lead a wonderful life without having to worry about having your bladder fulgurated or zapped with a light. So what we did experimentally was we took CFT073, again, a pathogenic strain of urosepsis, and coated it in a metal organic framework. So like that tobacco mosaic virus, what you see here are coatings of CFT at ZIF. So the bacteria is inside and coated in this crystalline material. This is distinct from what ZIF-8 looks like normally. The normal crystalline ZIF-8 that didn't grow on anything is on the right here, and you can see just large rhomboidecahedral crystals. Um, if we look at the characterization, the surface coating of ZIF is crystalline, and that matches up perfectly with simulated and pristine samples. And you can look at the distribution of elements over the surface, and you can see that essentially everything's all there, as you would expect. Now, what we found is in terms of degradation, we wanted to explore this in vivo. And in order to visualize this, we actually made the bacteria themselves glow red. So if it wasn't clear, the reason we used a near infrared Psi 7 dye in our detection modality is because this is a very good window optically for light to pass through tissue. And so what we did was we created a near infrared glowing bacteria that we could see through the skin of the mouse. Now, whole cell inactivated vaccines have been tried, and they are typically inactivated in two different ways. One, they are cross-linked with formalin, which essentially kills the bacteria and blocks all the structures in place, or they're heat inactivated. They're essentially killed with heat. And to make this simple, we created a formalin fixed version of CFT, which we call CFT fix. We essentially took the bacteria and added formaldehyde or formalin to it to cross-link it and killed it. And we directly compared it to the encapsulated ZIF form. And then when we inject the fixed formalin fixed bacteria, or what I will call the traditional vaccine preparation method, that injection lasts in the injection site for 2 hours. So, of course, your immune system is particularly well engineered to attack bacteria that invade surfaces. And as you can see, it does a good job. It gets rid of infection or what it thinks is an infection in 2 hours, which is entirely too fast for a prolonged immune recognition process. Now, the CFT at the CFT at ZIF, you'll notice, lasts significantly longer, over 5 days. And that's an important feature. So essentially what you have now is a slow releasing depot of your injection. And this is important because it's going to provide some time for the immune system to respond, which of course is why you have follow-up injections or booster shots, is to keep introducing this stuff to your immune system, because the injection normally leaves much faster than a normal infection timeline would allow. Now, I had mentioned the kinetic lability before, and this is sort of the re-emphasizing this, is that these are the tobacco mosaic virus. You can see here, these were actually removed from a ZIF. You can see they're actually structurally pristine, completely unharmed. What we also found actually is that in addition to chelators like EDTA, phosphates and albumin and protein are very good at removing the zinc. And they will completely strip the ZIF away, but more slowly, much more slowly than EDTA. And so essentially what you have is biological anions or macromolecules coming and disassembling your external polymer. And so if we compare this, and we can look at this using the tobacco mosaic virus as a second example of something. If we just inject tobacco mosaic virus, this is much smaller. The immune system is not as keen to recognize it. It lasts in the injection site about 2 days. If we wrap it in the ZIF, you can see again, this one lasts longer, over almost 9 days. And so again, we can show that even with the infection, even with a virus, we can actually retain them in tissues for much longer. And here is a radiant deficiency to essentially illustrate the very large difference in retention time. So again, showing a depot effect. And the reason why this depot effect is important physiologically is because of these things called germinal centers, and essentially they produce long-lived antibody secretions. And what happens is that when you have an infection, the local site, it's drained to a lymph node, and that lymph node develops this germinal center, which are divided into dark zones and light zones. And essentially, uh, antigen is brought in, and B cells in this thing are trained or selected for their ability to recognize the epitopes of the infection. And if they do recognize it, they go to the light zone where they are then essentially tested with the antigen. And if they bind, then they're allowed to go back in the dark zone to undergo what's called hyposomatic mutation or somatic mutation to essentially try to find the best antibody for that antigen. And so this is sort of a smart, a system within a system that's designed to select for the best antibody, but it takes time. Okay, it takes about a week or longer for the system to actually come up with the strongest, most robust antibodies that you can come up with. Um, these are then sent back into the light zone and then continuous selection. And whenever an antibody doesn't bind well or binds worse, that B cell is destroyed. And so ultimately, you end up refining only to B cells that have the highest affinity. Now, going back to our story on the cellular or the bacterial infection, what we did was they put them inside of a ZIF. And when we did this injection and we looked at endpoint titers at day 21, our CFT at ZIF was already way higher in terms of total antibodies than the fixed version or the heat-treated version. We also benchmarked it against that. At day 42, it was order of magnitude. So we had 10 times more antibodies from our formulation than the more traditional formulations of the fixed and the heat treated. And that bears out in terms of not only in the lymph nodes. I'm going to actually direct you to the lymph data over here on the right. What you can see is that T cell, and so the blue is the fixed formulation, and the green is the MOF formulation. In the local lymph node, T cells are roughly the same. Much higher, obviously, than baseline, but roughly the same. What's different, though, is the total B cell production, and that's significantly higher in the lymph node. But it's even more telling that in the spleen, which is indicative of a more systematic immune response, they're all higher. So we have much higher B cell, T cell, and CD4 and CD8 production. And so the immune system with the ZIF formulation is clearly way more immunologically active. And we can confirm that by looking at the germinal centers. We can look at the B cells, and we can see they're higher, and then separate subsets of B cells and the ratio of how big the germinal centers developed. All this data shows that the ZIF formulation actually is vastly superior, or at least editorialized, superior than the fixed and the heat-treated formulations. And this really is borne out in the survival data. So once these animals have been inoculated, what we then do is then challenge them. And so we give them a lethal dose of that pathogenic bacteria, and we see what happens. So completely naive mice are dead within a day. They cannot fight off this infection. The fixed and the heat treated both do fairly poorly. So you do get some prolongation of life, but not significantly. Whereas in the case of the ZIF encapsulated formulation, we essentially see a very, very good survival, something that is very hard to do. You can see basically, you can look at the bacterial loads in the mice, and it shows three of the mice actually resolved an infection. Again, this is something like CFU of 10 to the 9, so a huge bolus injection that starts up around here. In many cases, the saline, you see it's much higher because, of course, the bacteria is reproducing. And the fixed and the heat-treated versions are, they're more or less stagnant around where we injected, maybe slightly higher, but only in the ZIF formulations do we see obvious and significant reduction in total bacterial burden compared to what we injected. Now, Tuberculosis is another thing, right? Tuberculosis is, of course, a big problem in the developing world and a big problem in areas, but it's also a problem that's emerging once again in the United States and other so-called developed Western countries. And the truth is it simply has never left. The tuberculosis continues to be one of the deadliest pathogens in the world. Second, it's certainly the most deadly bacterial infection in the world. So it's pretty tough to treat and pretty tough to cure. Um, as an example, every single day, 4,000 people die from tuberculosis, a fairly large number, and 23,000 people will develop it. So it's a fairly significant problem globally. Now, what we found is that, as you can see, the ZIF contributes to the immune response, either through a depot effect or other biochemical pathways that actually make the immune response stronger. And you can see what happens is the injection. So essentially what this slide is that if we inject it, the moths break down. So this is just moth injected in an animal thigh. We remove the depot after 24 hours, and you can see that essentially the metal organic framework degrades, as I told you. We also note, though, that if we look at the lymph node, the draining lymph node here, this is panel C, we see a significant amount of zinc that's inside of that lymph node. And if we take that lymph node out and look at T cells, we see that the T cells themselves have become active. Now, there's no antigen in this. This is just pure ZIF. And so just the ZIF by itself seems to be priming the immune system, which is interesting. And it made us wonder, if it was the metal or if it was the ligand. We really weren't quite sure. And there's a lot of contradictory reports about exactly how zinc mediates the immune system. It definitely does, but how it works biochemically is still an area that's somewhat open to discussion. But what we wondered, though, is if we could actually tune this response using a metal that is a bit more well characterized in terms of how it can activate the immune system. And there is a pathway inside of cells called CS sting, and this is this is an inflammatory response that is activated by extranuclear DNA. So when DNA gets into the cytosol, it shouldn't be there. The cell responds by creating an immune response, and essentially that it becomes activated. And what people have seen is that manganese will turn this intracellular immune system on. If you add manganese to a cell, what you will end up with is these expressions of on dendritic cells that show activation. Okay, so activation can be done very specifically using manganese. So we decided, what if we developed a metal organic framework that uses manganese as a specific way to activate this sting type pathway? And so we used this concept of ZIF8, except we substituted about 50% of the zinc for manganese, and we loaded it all together. But just to further enhance it, we use this cyclic dipeptide, and this cyclic dipeptide also enhances or activates sting. So if the manganese weren't enough, we could also use CDN. We can see how they synergize, which they should, and we should be able to get a good response. One thing to note is the cyclic dipeptide does not enter into cells very well. It's very negatively charged, and so getting it into a cell is hard. It's also enzymatically easily degraded, so it's actually a pretty bad therapeutic. It has been in clinical trials. It's not worked very well. So a different approach is clearly needed, but it's the gold standard for reference. So we used it. Cytotoxicity, if we look at the combined metal organic frameworks, and I should mention, because cytotoxicity is often brought up, the metal organic frameworks that contain zinc, so ZIF8, these are less cytotoxic by a small amount, not large, but it's less cytotoxic than alum. So alum is already used in vaccines. The dose that we use is typically around the 20 microgram per milliliter, so well below the cytotoxic dose. And this just points out in this particular case that the mixed manganese ZIF is slightly less toxic than pure zinc, presumably because the reactive oxygen species that the zinc probably produces is mitigated by the manganese in sort of like a SOD-like reduction pathway. Again, we show some uptake experiments, this time with cells. And if we put a dye inside of the moth, we can see where it goes, and we can kind of see that um, we actually don't have particularly great overlap with the lysis tracker and the manganese dye, which sort of suggests that it gets out. The manganese, that the moth does not just stay in the lysosome, it does degrade and come out. So the metals should be able to get into the cell. And then we did cell uptake assays to essentially show that these particles are actually getting into the cell in the first place. Now, what we wanted to look at here is does this actually activate the immune system? And so we first looked at activation of dendritic cells. And, dendritic cells are sort of the unsung heroes. Everybody talks about macrophages as sort of the frontline defense, and macrophages just kind of are dumb and they eat stuff. But they do present antigens. But dendritic cells are really their job is to go find infections and try to work with the rest of the immune system to launch a defense against it. So activating the dendritic cells is very important. And just zinc by itself doesn't really do much. CDA, that the sting activating material, doesn't do stuff. The ZIF doesn't do anything, largely because it's not really immunostimulatory for dendritic cells per se. The CDA just doesn't get into cells, whereas the manganese, you can see, does activate the cells, as does lipopolysaccharide. The 50% manganese ZIF does significantly better. And when you add the cyclic dipeptide to the manganese as if it synergizes and you get much higher dendritic cell activation. And we can show sting activation using this Western blot. Essentially, we can show phosphorylation, which are these dark patches here, of key proteins that are involved in the sting pathway. So if they're phosphorylated, then the sting pathway has been activated. And you can see that with magnesium chloride, you get these sort of same bands as well as ZIF. Now, of course, you can't inject manganese chloride into a person. It's neurotoxic, so you have to be able to keep them all combined into a nice little nanoparticle so that it doesn't just diffuse everywhere and cause general systemic toxicity. Now, if we expand on this, so I already showed you this activation of dendritic cells, we also have production of TNF alpha and IL-6. With these, essentially what we show is that we do have a production of these inflammatory cytokines, but not as much as lipopolysaccharide. So if we had stuff like lipopolysaccharide, then pro-inflammatory cytokines would be so intense that they would make you extremely sick. And so we don't see that sort of large response, which is good, just in terms of safety. So we're going to activate the immune system. Then we wanted to test it in a tuberculosis subunit model. And so CysD Ag85B is a clinically investigated antigen for tuberculosis. It has been looked at with CpG, which is I think it's FDA approved at this point, adjuvant that helps stimulate the immune response. And then Advax, which is another one that helps stimulate the immune response. Now, these are selected specifically because this promotes an intracellular style immune response, what's so-called Th1 response, which manganese and sting activation do. So in our the UTI example, what we saw was what's called a Th2 or extracellular response. We were really interested in antibodies, and antibodies are not particularly helpful for tuberculosis because tuberculosis generally lives inside cells and antibodies can't get them. So what we need is a different kind of immune response that focuses more on T cells, not B cells. But the ultimate measure, of course, is how well they do in terms of log reduction of an infection model. And you can see BCG. So this is unvaccinated. You get 5.5 CFU to the lung. BCG does a great job in mice. It does not do well in humans. But you can see here that when you add this clinical Advax, you get a reduction compared to the unvaccinated. And that's really what we're looking for. We're not looking necessarily to compete necessarily with BCG, lower log reduction, that would be very promising. But on a mouse model, our initial assay is simply efficacy because we have to move to different animals that are more representative, including large primates, which is going to be better, or guinea pigs. We did an infection in vivo. So essentially we did an initial inoculation at week 4, and we looked at some cytokines that are associated with activation that we wanted to look at tuberculosis. And essentially what this data shows is that it does a really great job. You can see BCG down here, these little The little bright neon green actually doesn't induce that much cytokines, whereas ours stimulate a very strong, this intracellular type immune response. And so that's great. It suggests it should work. And then we challenged it. We gave the mice, actually, this was done by Dr. Angelo Izzo of the Centenarian Institute in Australia. We don't have a class three biosafety room to work with live tuberculosis like he does. And so there's live tuberculosis. And essentially what we see is BCG does great, as one expects, but compared to saline, ours actually does pretty well too. It does, in fact, reduce the total CFU burden, and so we get some adjuvant effect, which is very promising. So this suggests that this is possibly an effective adjuvant and a possibly novel adjuvant. Now, I did mention, and I'm going to go really quickly because we're about to run out of time, but our delivery methods to avoid needle um, have progressed a bit. So we're interested in non-syringe or non-injection methods. And so microneedles and stuff like that, these are published and we've looked at microneedles and stuff like that. And microneedles, essentially the idea is to get the vaccine into the skin because the skin is lots of immune response, lots of immune cells as the outer barrier of your body. So that's where your immune system is going to be the most active. And in the old-timey days, they had these multi-use nozzle jet injectors, which essentially shot a spray of the liquid into the skin, and it hurt. It spread hepatitis B, and they have been phased out. And the big problem, of course, is that it shoots a liquid, and the liquid gushes back out onto the injector nozzle, and that spreads disease. So we came up with a bit of a different idea. Instead of using liquid under pressure, we just shot it with high intensity gas. And so I had a student that came up with this little device here that essentially you load the MOF-coated vaccines inside of it and reassemble it, and then you shoot it into the skin. And what's great is that you can control it so that you can get delivery directly into the skin. So if we do this launching of the skin, we have the epidermis. That's not a good place to deliver anything. All the cells are dead, and so that's what we've labeled E. Dermis, which is live tissue, but not necessarily the best place to start shooting stuff. You want some in the dermis, but subcutaneous is essentially where you have a lot of the immune cells that we want to target. And when we use air or carbon dioxide, you can see we actually get a lot of loading in the subcutaneous and even in the dermal region, which is great because that's essentially a good place to do deliver some vaccines. Anyway, that's my talk. It was really lovely giving a presentation. I'm appreciative of everybody. This is everybody who contributed and the funding agencies that allowed this to happen, and I'm happy to take questions.
Thank you very much for your amazing lecture. So, dear colleagues, do you have questions for speaker?
Yes, may I start?
Yeah, sure.
Hi, Jeremiah. Thank you once more for accepting our invitation to participate in the Kharkiv Chemical Seminar. It's a pleasure for us, and it's nice to see you again. Thank you for this very nice talk, very informative and saturated. I was really impressed by this MOV-based vaccine. And looks like it's not cytotoxic, and it showed a very nice result. So my question is, what are like the major challenges for translating MOV-based this vaccine to the clinic?
Sure.
Yes.
So would represent an entirely new class of material from the FDA's perspective, and that is a huge burden to prove safety and efficacy. Aluminum oxide and stuff like that, that's been in the clinic, and small derivatives like that make it a bit easier. But metal organic frameworks would be new. So the barrier is essentially cost and effort. As far as we can tell, it definitely seems effective, and I think we are actually at a point where we can move to larger animals, and even we are trying to write proposals. Although funding for vaccines has been incredibly difficult now these days in this country, but something that I think is possible. I think we can move towards translation of this. I think the results are fairly impressive and or at least good enough to substantiate moving towards um, a larger trials.
Yes, I completely agree that it's very impressive, and I wish you good luck with this. And maybe one more question about the first part of your talk when you told us about small molecule molecular probe.
Hmm.
You used this for diagnostics. And before the second part, you showed us like one slide with photodynamic this catheter. But what about photodynamic efficiency of this probe? Am I correct that we can use it not only for diagnostics, but also for theranostics?
Yeah, that's right. So the probe that I showed you doesn't have much in terms of photodynamics, so that's light converting it into reactive oxygen species. We have different designs for that. But in terms of photothermal, so the ability of the dye to heat things up and kill cells, that works pretty well. We can heat the cells up to over 45 degrees with stained cells, and that's enough to cause cellular apoptosis, which should essentially that kills the cells, releases the bacteria, or it actually flags the cells for clearance by the immune system, which actually is even better because that way the immune system comes and clears it out, and you don't just eject cells into the bladder and for further spread.
Okay, I understood. Thank you. Thank you once more for this nice talk. Thank
Thank
you.
you.
Thank you, Alessia. Thank you, Jeremiah. Okay, dear colleagues, more questions?
May I ask a question, please?
Sure, please.
Thank you very much for your very interesting talk. I dealing with the synthesis of inorganic nanoparticle, and up to now there are a lot of questions, a lot of literature, and a lot of work devoted to the antimicrobial activity of such inorganic nanoparticle, metal or maybe metal oxide. There are a lot. They also propose a lot of mechanism of such antimicrobial activity, including ROS production and metal release and so on. And in your opinion, is there is any future of this approach to use inorganic nanoparticle to fight the antimicrobial resistance problem?
I think
Because
so.
I
Yeah.
think it is very interesting topic to be studied.
Yeah. I think so. I think some metals are obviously going to be better than others. I think zinc is okay in terms of antimicrobial activity because it can clog up manganese transporters in the bacteria. Manganese is not particularly good because it can stimulate bacterial growth. Iron is a bad idea because it really can stimulate bacterial growth. Copper is great. Copper is really antimicrobial as a silver and all those sorts of things. I think in the context of linings and coatings for catheters and other things like that, or necrotizing fasciitis and other skin surface and skin diseases, there's a lot of opportunity here for inorganic nanoparticles that can essentially shed small amounts of reactive metals like copper or silver to and essentially sterilize equipment or keep tubing and other medical equipment clean and clear. I think that there's certainly an area there, and I know that area is actually fairly well explored, and a lot of good research has come out of there.
What about nanoparticle as a therapeutic agent, not just coating or something like that? I know that there are there are a lot of problems with approval of this object for medical application. It is very cost and it has a lot of question in Europe, not just in Ukraine, but in Europe, in America. But I think this object could be studied and used for such application. because even in our laboratory, in our group, we have a very promising result concerning antimicrobial activity of nanoparticle, not just as a source for metal ion release, but nanoparticle as a whole therapeutic system.
Yeah, I think that that certainly would, if you're specifically interested in transport, because nanoparticles, well, metal ions themselves are fairly tightly regulated in terms of cellular uptake. So if you want to get metals in any appreciable concentrations inside of cells in a way that doesn't kill the host, it has to be in a nanoparticle. Or at least in some kind of other targeted system, because you can't just inject copper salts into people, right? Or other metal salts. This is very dangerous. But in making a nanoparticle, especially targeted nanoparticles that can recognize infected cells or infections, I think that this is probably the actual way to go. If you're focused on nanomedicine, because like you probably know, and like I said, um, just indiscriminately putting metal salts on things just won't work. You need to have some sort of delivery system.
Thank you very much. Thank you.
Thank you, Svetlana. So, colleagues, maybe more questions? I don't see hands or microphones switch off.
Okay.
Okay. So your lecture today gathered together a lot of scientists, not only from chemistry and biochemistry, but also physicists, biologists, material science, and I hope all these people join with your lecture. It was interesting for them. Thank you again for accepting our invitation and for very, very interesting lecture.
All right. Thank you for your invitation. I appreciate
you.
the opportunity.
Thank you. Thank you.
Thank
Yeah,
you.
thank you, Jeremiah. Thank you.
Yeah,
Have a nice day today.
Thank you.
I will.
Very nice lecture.
I appreciate it.
Thank you.